So this is the paper that I was telling you guys about. This one is titled Explaining the Impact of Mutations on Quantification of SARS CoV-2 in the Wastewater, where the COVID concentration in the wastewater is estimated by the N1 and N2 proteins using quantitative PCR. This is a specific type of methodology for detecting COVID in wastewater, and it noted that this started to diverge in 2022, around July of 2022, from different sampling sites in Japan. So this might be slightly different to the US, but it's good to note that this is something that started to diverge in July of 2022, where importantly what they noted is that due to two mutations in the N1 probe region, it resulted in an underestimation of viral concentration levels. And this is because what we do currently is we had assumed that the N1 to N2 is going to be stable and it's in a 1-to-1 concentration. And we know that the N2 protein is stable, or it has been stable so far, but the N1 protein has been slowly gathering mutations, and because of this, it's resulted in an underestimation of the wastewater values, which we'll show later in this paper. So first, let's give a quick overview of why wastewater is important and why we use it for COVID detection now, as opposed to case-based COVID detection, where you go to the hospital, you get tested, and then you get a result. Well, first, it's because it's really easy to do in a way. Everyone poops, everyone pees, all that waste goes to the wastewater system, so it all aggregates at one location within your immediate area. Obviously, everyone's waste goes different places, but it goes into your immediate area in one location, and from there, you're able to sample that as opposed to going out and finding individuals that are sick or potentially sick with COVID. So it reduces like the man time or the man hours of needing to track all these people down, or to have everyone testing and refusing tests or not going to the hospitals when they're sick with COVID to get tested. So it's like this inadvertent direct way to get measurements to see when people are actively sick with COVID. So in short, it acts as a good community surveillance method without having to test individuals. You test populations. Now, moving on. So now how do they actually measure this COVID in the wastewater? Well, they use something called quantitative PCR. And this uses target primers and probe regions of the protein itself. So this would be the N1 receptor binding domain and the N2 receptor binding domain of the nucleocapsid protein, which is just regions on the protein. So just like specific areas, these primers target to it and then bind to it. And then when they've targeted it to it and binded to it, then it usually will be detected with some sort of fluorescent detection. But the problem is if you get mutations on these regions and they're significant enough so that the primer with the tag on it doesn't bind to it, then it's not going to detect COVID. It's not going to fluoresce and show that COVID is existing in the solution, even though the COVID is there, because the primer is no longer well matched to that receptor binding domain on the COVID. And that's because the COVID has mutated sufficient enough so the person would have to develop new primers to match to the COVID in the wastewater. Therefore, as highlighted in the blue here, genome mutations in COVID itself impact the target regions for the qPCR analysis, the quantitative PCR analysis, and can influence what is actually being measured. And this reduces the accuracy of the analysis itself. However, the authors are saying not enough attention has been paid to this issue. So far, it's been overlooked largely and hasn't really been corrected for in our current COVID wastewater analyses and public displaying methods comes in. During the second half of 2022, so around like July of 2022, the authors in this paper noticed a discrepancy in the COVID concentrations in wastewater based on measurements from the CDC due to mutations in the N1 and N2 regions, suggesting potential inaccuracies within the data. The authors noted that in the beginning of the seventh wave in July of 2022, until then, the ratio between the N1 and N2 concentrations had been close to 1, like a 1-to-1, which is what we assume, but it declined significantly after this, where the data noted a 1-to-2 order of magnitude lower concentration based upon the N1 region than on the N2 region, and since it's N1 divided by N2, a lower N1 with a same level N2 is going to result in a lower measured concentration, even though the true value would be higher. So what do I mean by that? So the measured concentration is what you get with your machines, but the true value would be what the value actually is. So your measured value is being less than what is actually there. That's what it means. And while the magnitude of this divergence was not constant across labs or sample preparation methods, the timing at which it started was nearly identical across all datasets, which means that different processing methods are going to have different impacts on this, and that depending on the concentration of the site, so like that is a good indication that, oh, a variant is what caused this, a change in the variant of COVID is what caused this. That's what people start thinking when you see that kind of information. And then we have confirmation of that in this paragraph below. So what happened during that period of time in July of 2022, while we're switching from the dominant Delta sublineage to Omicron sublineage. So we're having that big jump from Delta to Omicron that occurred between January and March of 2022. Where, interestingly, in that jump from Delta to Omicron, all of the major mutations were in the N1 probe region. So they're all focalized on that N1 binding region of the nucleocapsid protein. And this resulted in fluorescent signals that were weaker than the wild-type sequences. But interestingly, the DNA amplification was not significantly impacted by this change. However, the fluorescent intensity was, and the fluorescent intensity is what is being measured for the COVID concentration through this method. So even though the amplification, the number of amplifications is not changing, the fluorescent signal that is being measured is changing, and that's impacting what is being measured with the device. And this figure right here really demonstrates this issue. So when you have this mutation on the N1 protein and this mutation on the N1 protein, it ends up having about a 15% reduction in the fluorescent signal as compared to the wild type. And then when you have tandem mutations, when you have multiple mutations together, it reduces it by about 40%, which means that your measured value would be 40% lower than what it should be, which is insane. So what the authors have definitively shown through their work is that these specific mutations in the N1 binding region have decreased the fluorescent signals in quantitative PCR, which leads to underestimation of the virus concentration when calculated using these quantitative cycle threshold values. Where again, the reason this is such a problem is because to date, the N1 to N2 concentration is expected to be constant at 1. So the authors are demonstrating here that it's important and very helpful to monitor the N1 to N2 ratio routinely to detect when it's causing these changes, and then applying a correction factor backwards to make sure that you're compensating for these changes. Where the authors emphasize 3 additional strategies besides routine monitoring. The first is emphasizing the need to target multiple genomic regions for each sample to make sure that if you're just like targeting just the N1 and N2 and you have these issues where it's causing some sort of split outside of a 1-to-1 ratio, then it's going to impact it. So making sure that you're potentially expanding the targeted range so you can compensate for these effects. They then point to 2 particular processing methodologies, the STM method and the CY0 method. And finally, the authors emphasize that you can't really anticipate future mutations for COVID. You can only basically respond to them. So addressing this issue analytically as opposed to speculatively going outwards, and accommodating and adjusting for the effects of the mutations backwards, so doing post corrections, which is what I talked about earlier, offers a more robust and practical way for handling the effects of these mutations on the quantitative PCR data and thus our COVID wastewater monitoring.